Functions. The potiometer is appropriate equipment commonly used to measure the rate of water uptake of a transpiring plant. It may also be used to compare the rate of transpiration of different plants under the same or different rate in all the plants. Description and demonstration of a potiometer. The potiometer consists of a wide-mouthed bottle filled with a stopper bearing 3 holes. A dropping funnel with a tap is passed through one hole. The stem of a freshly out shoot is passed through the second hole. The stem should be cut under water so that no air bubble enters the xylem cells. A long bent glass capillary tube is passed through the third hole. A suitable graduated scale is attached to the horizontal arm of the capillary tube by means of rubber bands. The bottle is filled with water and stopper carrying the glass tube. The funnel and the chute are placed firmly into the mouth of the bottle. The dropping funnel is also completely filled with water. The tap is then opened to allow to flow down the length of the funnel so as to fill the bent capillary. And while this is being done, an air bubble is trapped near the free end of the tube. The top of the dropping funnel is then closed. While transpiration is taking place through the leaves of the chute, water is absorbed from the bottle by the stem. As the volume of water in the bottle decreases, the water column in the capillary tube is down into the bottle and the air bubble is seen to move towards the bottle. By looking at the graduated scale, it is possible to read off the distance the air bubble has moved. This is directly proportional to the amount of water lost by capillary tube as a result of the water taken up by the chute. If a graduated dropping funnel is used, it would be possible to measure the amount of water absorbed by the chute. This could be done by refilling the capillary tube with water from the dropping funnel and reading of the amount of water required for this. If the experiment is carried out over a fixed period of time, the rate of transpiration, water uptake, can be calculated. Volume equals pi r/h, where r is the radius of the bulb, h is the distance, v is the volume, and pi equals to 3.142 or 22/7. Module 2. Animal Physiology. Unit 1. Measurement of heartbeat and effect of exercise on heartbeat. Lesson 7. Experiment to determine heart rate and beating rate. Introduction. During blood circulation, the heart pumps the blood round the body. The rate at which the heart beats is referred to as the heart rate or pulse. Blood supplies the body nutrients, oxygen, and other necessary materials for proper functioning of the body. In the same way, the breathing rate is the number of times per minute the diaphragm moves up and down. By breathing, the body through the blood is supplied with oxygen for respiration and carbon dioxide is removed. Thus, determination of the heart rate and beating rate is vital equally. The effect of dynamic exercise on both heart and breathing rates are also considered. The determination of the heart rate and breathing rate is vital equally. The effect of dynamic exercise on both heart and breathing rates are also considered. Materials: stethoscope, 1 per group; stopwatch, wristwatch. Procedure: 1. Put the head of the stethoscope between the 4th and the 5th intercostal muscles at the left hand side. At convenient time, Start the stopwatch and count the number of beats per minute and record it. 2. Heartbeat. Work in group of 5 or 6. 2 members, A and B, will act as the specimens. Specimen A will sit relaxed, his left hand rested on the table with palm up. Other 3 members of the group in turns will read the heart rate of specimen A. Using this method, put the 3 middle fingers of your right hand on the wrist of the specimen A to feel the vein. Feel the pulsation. At a convenient time, start the stopwatch and count the number of the pulsation in a minute. Find the average of the 3 numbers. This should represent the heart rate for specimen A. Repeat the same for specimen B. Collect all the heartbeats for the females and males separately. Find the mean and deviation. Check the normal heartbeats for both male and female. 3. Breathing rate. Retain your groups and specimen A and B. Observe the breathing of specimen A and B. This can be done by placing your hand on the side of the chest and observe the up and down movement of the thoracic cage. At the convenient time, switch on the stopwatch and count the number of movements up and down in a minute. Put together the breathing rate of all the members of specimen A and of specimen B separately and find the mean and standard deviations. 4. Heart and breathing rate and exercise. Retain your group to carry out this section of the lab work. Specimen A should run about 200 meters and his or her heart rate should be taken quickly by only members of the group. Treat the class data as in above. Specimen B also should run about 200 meters and the breathing rate taken quickly by a member of the group. Treat the class data as in 3 above.